acid.
You talk about plasmids?
I think so, yeah.
Oh, the plasmid one, you just have to go to, so there's sequence map, linear map, plasmid map. I was just showing you the plasmid map.
Oh, okay. I thought we were doing
Yeah.
something with it.
No. You can look at the plasmid map, right? This is how your plasmid is supposed to look after this insertion, right? Sorry, where am I? Okay. So you should have this. Now, since you have mastered the technique of building your plasmid, here is an assignment for you to think a little bit, okay? Let me zoom in. Okay, so we have done basically all of this, all right? Now, Susie has some dilemma, okay? I'm being a mean PI, so of course I will give you some things to do. Now, Susie would like to express this gene in P. cambia 1301 because she wants to do the Gus assay that I showed you guys to see if the protein is being translated, okay? How would you modify the sequence to express the Gus protein? All right. Hint, hint. So, if you go to Pcambia, all right, if you look at the plasmid for Pcambia, right, the Gus protein is here. You still require the intron for the Gus protein because it has to be expressed in plants. So these two are important. Make sure you don't cut these. There's only two sites, NCO1, BGL2. All right. You'll need to choose which one is most suitable for you to insert your sequence. Your sequence has a stop codon, meaning if you insert it into this area, if you insert it into this area, you will need to remove your stop codon so that translation can continue to produce the Gus protein that should be expressed in plant. Okay, so that is Susie's dilemma. You will have to I'll be right there. You will have to figure out, okay, how to remove the stop codon so that translation can continue. Because if you have the stop codon on your sequence, it will not continue translating to the Gus protein. Okay, that's your hint. Also, NCO1, I don't know if they'll show there. NCO1 is tricky, okay? Because the restriction site for NCO1 is CCATGG. If you notice, it has an ATG, meaning the polymerase will not bind to your ATG, it will bind to NCO1's ATG, meaning it might change the sequence, the order of your sequence. So you need to modify your sequence to ensure that even if the polymerase bind to this ATG, your other sequence will be read by 3 codons. Okay? So that's dilemma 1. Dilemma 2 is Susie wants to express a very short peptide. This is the peptide she wants to insert into the PES plasmid, okay? Right behind the galactose promoter. All right, let me just put that, galactose promoter. Okay? So, how will she need to fix this sequence in order for it to be properly expressed? So you need to make sure it has a start codon, it has a stop codon. You need to make sure it is in frame, meaning it can be read by triplicates. Okay? Only those two, see? Right there. Now, for you to, what I want you guys to do is when you are finished, So you should have 3 vector maps. One for this assignment that we just did, right? One for Susie's pCambia and one for her PS vector. So you should have 3 vectors. You will need to share these vectors with me. There is a share tab here, sorry. All right, so you could share this link, all right, with me. You turn on the sharing. If you want to tell me something like OMG or whatever, just share it. You could copy the link, all right, and just send me the email, okay? This is due. Myron say, Miss, this is a lot. Okay, you know what? Fine. If this is a lot, you just need to do number 5 then. Okay, number 4 is very tricky. This one. But you know, if you master this, you could go and do molecular biology like, okay? But this one, fine, I could do it as a bonus point. All right. Yes, so you could do the number 4 as a bonus. And then number 5, you have to do at least one of them, okay? But this is because you could hear me and we could listen, we could try work it out, but for you to really understand the process is to go in there and try and see, you know, that's why, you have to insert this, this, you know, and figure it out on your own. Is a good way to learn, like especially if you're, how do they call those people that learn by doing things, right? So if you're that type of learner, you would need to go and get your.
I got an error when I was trying to do the amino acid annotation.
That means that your sequence is not being read, is not in frame. So you would need to check. Did you start with ATG and end with the stop codon?
Yes.
You need to make sure that from ATG to your stop codon, you don't have an extra amino acid. And if you guys if something is giving you trouble, you can send me the link and then I can check to see what's happening with your sequence. So you can always share the link with me later via email or one of the message. Yes, submit individually. That thing no hard for check. I just do and I can know whether or not your map is accurate or not. Okay?
When did you?
It's due next week, Friday. But if you're having problems, for example, like what Edwin say, translation is giving him problem, which is a really bad thing. That means that the protein will not be translated. Okay? If it's giving you problem, let me know. And then I will see where I could help you between today and Friday. Kind of ish weekend because I need my rest too. But send me the link and I'll let you know if where you're going wrong or maybe you've inserted certain sequence. And then Yep. But Friday will be I will not help you anymore at Friday because that's when it's due. Okay? But before then on Friday, just send me a link. All right, guys. Any questions?
Okay, and one last one. The software is save all of our projects automatically, right?
Yes.
Yeah. Okay.
Mm-hmm. You could form different project folder too and stuff so you don't confuse yourself. By the way, I don't have that here with me. Your kinetics thing to work, right? Is it working, or else I'll just send you guys a GitHub for it because just download it yourself. Send it to my.
I prefer the GitHub. Try that at least.
You haven't tried it?
No, try that.
What?
As in try the GitHub.
Okay, hold on. Let me send you guys it. I think it's in tracked. Yep. So you can go to the GitHub. Let me send you right here. All right, so this is the this is where I got the program. What program, but just some codes. So you can download it straight from there, see if it works better than from the folder. And it should be able to run. My one's still running good, so I don't know what happened to you guys. But download it straight from there, then from the website. Any more questions, guys? I'm hungry. My brain can't think anymore. It's running out of glucose. Okay, if no more questions, then doesn't CTRLY work? You mean paste? Wait, no, cut. What is CTRLY?
Like,
Is it?
because I deleted a lot of my stuff by mistake.
Let me see. Well, you can always do it over.
Miss, who it on right now?
You can always redo it because sometimes you might. That's it. Oh, that's how you properly zoom in. Oh my god! And hey, there I was. Can't help you there. See, that's why I say it's best if we go in a classroom, then I could see what's going on with you guys. Because I could only guess what's happening in those plasmids right now. So I would say if it's getting really confusing, do the whole process over again. It also helps you to solidify it in your brain anyways. So sorry Edwin. Okay, any other questions? The clouds got dark. I don't know if there's any videos that good. Anyways, if anything, next week you can come. I'm right here. Who is messaging me now? Oh, okay. Someone has sent me a link, but I'm not gonna look at it, guys. I need to go eat because I'm starving, and I won't be able to help you if my brain is starved. So see, they're all leaving already. So thank you, guys. I will see you next week. If you have questions, just send me your link with your question. Don't just send a link because I will assume you don't finish and you didn't send it to me. So send the question along with the link if you meet any kind of trouble building your plasmid. All right, guys. I need to go now. I'm hungry. And I'm hungry when I'm hungry. All right, guys. Bye. Have a good weekend. Study. La la la. Bye.